Hello everyone, welcome. Today I'm going to talk about our recently published study. Some of the intriguing questions I was motivated for this study includes how did first cells assemble from prebiotic components and what fundamental physical chemistry underlies living cells. This study is going to be about prebiotically relevant compartments and how their components determine its functionality. Why do I care about compartments? Because all cells are compartmentalized, and cells use both membranous and non-membranous organelles to compartmentalize biomolecules and regulate reactions, which is important for controlling cellular activity necessary for life. And here are listed advantages of compartmentalization. For instance, you can maintain high local concentrations, you can have selective entry and exit of solutes, and you can provide favorable conditions such as pH and different concentration for crowding agents, and you can protect inside from the damaging conditions. There are two ways of possible prebiotic compartments, including membrane-based and liquid droplets. You can have your liquid droplet assemble into these compartments and use your biomolecules. You can have your biomolecules compartmentalized in these little droplets, or you can have your lipid vesicle, and you can actually load these lipid vesicles with biomolecules. As you can see here with primordial earlier soup, you have some molecules that can actually form both of these compartments. I'm going to focus on phase separation specifically for this talk. I'm going to focus on specifically associative phase separation and as complex conservation. When you have negatively and positively charged molecules and when you mix them at certain ratios, you will have conservate droplets, condensed conservate droplets, and you will have most of your molecules condensed in this droplet phase, and you will have polymer deficient continuous phase. These two phases exist in equilibrium. Even though phase separation and condensation has been recently gained attention, the first this term coined in 1929. Let's say you have these periodically relevant molecules. They are charged and they are negatively and positively charged, and you mix them. What you will observe is either this clear solution, or here on the right you will see as a turbid solution. You can basically differentiate by eye, but you can also use UV visible spectroscopy differentiate between these solutions. So if you have low turbidity, you will have high transmittance and the light will pass through easily, and if you have high turbidity, you will have low transmittance and light will not be able to pass through easily. Of course, when you mix them, and they will look like this, but you don't know actually what you have in your solution. If you have this clear solution and low turbidity solution, it will look like this under the microscope as your solution, and you're not going to observe any formation. However, when you have the high turbidity, you can either have conservates, liquid conservates appearing as spherical fluid droplets, while precipitates form these amorphous solid clusters, and you will need microscopes to differentiate between this. I really like this structure. I really like this picture because it does show how phase separation can be affected by just simply changing the salt concentration. As the title says, polymer length and salt affect the phase separation. Here you can see the x axis, the salt concentration, As you increase the salt concentration, you can change the aggregate formation you see here, like big chunk, to conservate, and you can change the conservate to uniform solution, even more increasing the salt concentration to higher values. So other important factors also include that affect that phase separation includes total polymer concentration, polymer length, and charge density. It is quite an interesting phenomenon that can be affected by many different parameters. And recently people discovered that activities in cell can be compartmentalized by phase separation. Number of cytoplasmic and nucleoplasmic cellular condensate composed of RNA and protein have been reported to have liquid phase characteristic. Non-membraneless organelles within the cells, such as nucleoli and P granules, have recently been found to exhibit liquid-like behavior, including spherical shape, fusion, and dripping. There are many consequences of this, such as biomolecular partitioning, colocalization, sequestration, and regulation of metabolism. So, my motivation was for this study just to answer a couple of questions, as I mentioned at the beginning of the talk. How did first cell assemble from prebiotic components? There are multiple questions underlies. Within this question, you can actually ask further, such as, can we form compartments with low multivalency components? How low we can go? And another big question that motivated this study is what fundamental physical chemistry underlies living cell and protocell models, and how they are connected. Here are the molecules I have employed. I have used positively charged lysine and arginine, and negatively charged glutamic and aspartic acid. Number of charge per molecule and length play a crucial role in phase separation. Longer the molecule or higher the charge density, there is higher propensity to phase separate. We were curious how short we can go and still able to get compartments. So we change the length from monomer to 100mer. And I also included nucleotides, AMP, ADP, and ATP, which has different charges. AMP has 2 charges, ADP has 3 charges, and ATP has 4 charges. Then I examined all this combination between the negatively and positively charged molecules, and of course with different lengths, and I just classify them into 3 categories. First one, if I didn't observe anything under the microscope, also supported by the UV-vis studies, I will call them no formation, and with this little empty square. And when I combine them and they form compartments and conservates specifically, I will call them conservate. If they just form these amorphous solid structures, and they will called aggregates. And colors above the pictures represents the condition observed for polyelectrolyte mixtures. Liquid conserves appear as small spherical fluid droplets, while precipitates form amorphous solid clusters. Both lysine and arginine formed conservates with ADP and ATP, as you can see from the phase diagrams just under the pictures. Only, however, arginine was able to form conservation with AMP with the lowest charged nucleotide. Arginine seems to have stronger interactions compared to lysine. Then I look at the possible combinations for amino acid combinations and observe some trends, such as Glutamic acid tends to form more aggregates compared to aspartic acid. Arginine contained composition could phase separated shorter lengths, but aspartic leads to more phase separated combinations than any other. So I had quite the library to explore physical-chemical properties of these compartments, right? I'm not going to talk about all the things we have done, but if you are curious, you are welcome to reference for the paper here and explore all the partitioning studies and pH studies we have done. So basically, I'm going to talk about partitioned nucleotides and their structures and the rest of the talk. When you add a labeled nucleotide, they do partition in the conservative phase selectively, and you can actually calculate this concentration by basically using calibration curves, and it will give you the value within the droplet. Of course, I had many systems, so I just have selected certain ones to further study, and they are selected here. You can see it has a different length, and I just increase the length, and I also choose one system with a different identity, including ATP. In order to understand the structure of nucleotides within the droplets, I use double-stranded RNA. I use two labels. One of them is donor and the other one is acceptor, fluorescent labels. And if they are close enough, you will observe energy transfer, and you will observe higher FRET efficiency. But when they are separate, there's going to be no energy transfer, and you will observe lower FRET efficiency. Depends on the distance between these strands. If you look at the FRET efficiency graph here, this is for different system. In x-axis you will see the different system corresponding, and you will have the corrected FRET values for each system. As a control, I have done single-stranded systems, which actually basically same strand with two different labels in a buffer, and as I expected, I observed zero FRET efficiency in the buffer. However, when I had double-stranded system within the buffer, this is going to be our base, and this is going to be the highest value we observe, and it was around 0.6. And what I observed is very interesting. As I increased the length, the FRET efficiency went to lower, which means that my double-stranded structures was melting as I increased the length of the components of conservates. So just to summarize, I have shown with this study that membraneless compartments can be used as RNA concentrating microvolumes. And actually, when you think about the shorter length components, they're actually more effective than their higher multivalency counterparts doing that. And the compartments can accumulate functional arrays while largely preserving the structures, which is very important to function. So when you have these shorter components, they will actually preserve the structure, which can help improve the functionality of the conservates. I just want to acknowledge the funding from NASA and NSF and my previous PI, Kristin, for the study, and also thank you for listening.